Very good. Thanks, Sean, and the thanks to everybody at Tandem and Red Thread for putting this together. Very excited to introduce Rakovina Therapeutics to you, which is a new venture that we are launching here. I'm joined on the call by my colleague Dennis Brown, who's a co-founder of the company, and also David Hyman, who's joining us as the Chief Financial Officer, and they'll be available to answer questions when we get to that section. We're very excited about this because compounds that we're developing have been in process for some time and are really now positioned to take advantage of a very new and exciting understanding of tumor biology that really is related to DNA damage repair, which, as we'll talk about, is somewhat of an Achilles heel in cancer cells that we can exploit to selectively target tumor cells while avoiding any impact on normal cells. Just a bit of an agenda here. I'll talk a little bit about the history briefly, just to understand sort of how we got here. Like I said, this is projects that have been going on for some time. The summary of the offering, overview of the company as we are today, and we'll go through some science after that very briefly, because I know there are a lot of folks on the call who will be interested in the science, and a number who may not be as well versed in the science as some of us. So we'll talk about the company and the overview first, and then we'll dive in a little bit to the science, but and then we'll turn it over for questions. So as I mentioned, this is a project that has been bubbling along for some time, and it traces its history back to about 2011, when a company in California called Therapeutics acquired a number of patents from a company called Kenyon Pharmaceutical. And Kenyon had funded a very prominent chemist called Wang Shen, whose name will come up a couple of times in the presentation here, to undertake some drug discovery. Wang was able to retain the ex-China rights to everything that they funded, which was the work that was done in his lab in California. Several dozen chemists worked on these programs. Those rights became a company called Therapeutics, which Wang founded with Dennis Brown, who's on the phone with us. So over the next several years, invested in advancing those candidates. And in November of 2018, Nugen was acquired by a company called Edison Oncology Holding Corporation, which I founded with Dennis at that time. We continue to develop the Nugen assets, and in particular, we're looking at opportunities around a class of PARP inhibitors, which we call the K2000 series. Earlier this year, we were approached by a CPC about an opportunity to spin out the PARP inhibitors, and we thought that was a very interesting idea, considering where the DNA damage response field has gone to, and these are sort of front and center as a next generation opportunity. And we spoke to a number of CPCs and eventually entered into an agreement with Vencero Capital Corporation in Vancouver. And we didn't want to just have the 2000 series, so we actually were able to build a bit of a pipeline around this opportunity with two other classes of compounds which fit squarely in this DNA damage repair opportunity space that were also invented by the same chemist, Wang Shen. So we've got a very nice suite of product opportunities here that we are looking to advance rapidly through this CPC combination into a public listing. The question will come up, why go public now when we are still relatively early stage? And the answer to that is simply access to capital. We know that we can take where we are right now and move rapidly toward clinical trials. When we get there, we want to be able to raise money quickly, and that will be available to us through the public listing. And you'll see the timelines as we go forward and how rapidly we plan to move. So this is a subscription receipt offering, $20 per subscription rate, which is a share and a half warrants. We think the valuation is very fair in terms of where the company is and leaves a significant amount of upside on the table. We're looking for a minimum of $2.5 million. There's well over $1 million committed so far, including from insiders. That will allow the business combination agreement to happen and the exchange to approve the public listing of the company. The funds raised in this round will be escrowed until the closing of the business combination agreement, at which time they will be free trading. So there will be no hold as soon as the exchange approves the business combination agreement once the funds are raised. And as I said, it'll be a share and a half warrant. Cap table will look something like this, where new investors coming in this round will own just over 20% of the company at the minimum offering, and obviously with additional capital in would own more than that. The Vincero shares, vast majority of them, and all of the Nugent shares that are being issued to the U.S. company, which is a subsidiary of Edison Oncology, those will all be escrowed. So the free trading shares here will be those in the subscription receipts offering, and happy to answer any questions on that. Okay, just to go through where we are as a company and what our goals are, is this concept of an Achilles heel, which is DNA damage response, is something that has come along in the biology research over several decades, but has really now just sort of come to the forefront to the point where a number of big pharma companies have directed efforts just in DNA damage repair, and we believe that where we are is at a forefront of that. DNA damage happens in our cells normally all the time, from radiation, chemicals, toxins, UV lights. It's a natural thing that happens, and our bodies are very good at either repairing that damage, or if it can't be repaired, you end up with something called apoptosis, which is a fancy word for cell death. The cells are programmed to die if the DNA damage repair function can't fix the cell. In cancer cells, you'll often have a mistake or a problem with one or more of the DNA damage repair mechanisms that lets a mistake or a mutation escape through without this programmed cell death happening. And that's where you lead to uncontrolled growth and tumor formation. And what happens when a cell has a defect in its DNA damage repair functionality, it relies very heavily on alternative pathways. And what we've learned is that if you target those alternative pathways, you end up with selectively killing the tumor cells. And that's a process known as synthetic lethality. And this is exactly what we're after here. So this is relatively new understanding of tumor biology that we can do this. And it's been successfully employed with the first sort of blockbuster drugs that are out there targeting DNA damage repair, which are known as PARP inhibitors. PARP inhibitors have been approved over the last few years. There have been a handful of them, 4 of them, in fact, that are approved by the FDA, generally all by big pharma. And this is a huge growth sector. These have now become standard of care, particularly in breast cancer, ovarian cancer, prostate cancer, where we know that there's a secondary mutation that can be targeted. So this idea of synthetic lethality is well understood in these tumors. The market is growing rapidly. Every single one of these approved drugs is expected to be a blockbuster, which means $1 billion plus in sales. And the space, just in PARP inhibitors, is expected to be nearly $9 billion over the next few years. An important thing to keep in mind is that these compounds, because they are so important and such major drugs, not only PARP inhibitors, but other DNA damage repair inhibitors, are driving acquisitions and very significant partnering. Medivation was acquired by Pfizer to get access to their PARP inhibitor in 2016 for $14 billion. And GSK bought Tesaro just last year for $5 billion, just to get after these compounds. This is how important they are. And this is where we're positioning the company to be able to really blossom and grow in terms of building value. Now, the PARP inhibitors have been successful, but the opportunity that we have as Rakovina is we now also know that they aren't that successful in terms of improving survival. So they'll stop the tumor from growing for some time, but the tumor tends to come back with a vengeance. And why? Because resistance will develop to these treatments because the cancer will adapt to them. And so next generation agents, like the ones being developed here at Rakovina, are very important for moving the needle in terms of improving survival. And you can see some of the publications here that reference this type of thing. So what happens in the cell is when you start on the left side of your screen here, you have a tumor cell that's kind of growing out of control in the blue here, and you inhibit it with you hit it with a PARP inhibitor, and the tumor will shrink. It's fantastic. These things work very well. But over time, you end up with resistance developing from a number of different mechanisms. Sometimes you have a cell in there that's resistant to the PARP inhibitor. You kill everything else, and it grows up uncontrolled and has a privileged environment. Sometimes the cell will upregulate mechanisms just to pump the drug out. And so we design drugs that are away from those types of pump mechanisms that aren't affected by them. Sometimes there are new mutations which will repair that alternative pathway, and other times the tumor just simply escapes via metastases to a place where the drug can't reach it, such as in the brain. None of these approved PARP inhibitors have very good brain penetration, so when a cancer cell can metastasize into the central nervous system, the drugs can't chase it, and that's something that's very important to us. So our vision is these next generation DNA damage response inhibitors that we have our hands on, that we're going to develop rapidly. Number one, improve the PK of just a plain simple PARP inhibitor, make it available to chase the tumor into the brain. This is game changing, and we believe we're at the forefront of this with the chemistry that's been developed by Wang Shen. We also want to be able to overcome resistance. So one of the things that we know that happens, for example, is that when you inhibit the PARP mechanism, there's another set of pathways that's upregulated called HDAC, which responds to the PARP inhibitor and creates resistance. Well, design a molecule that anticipates that and inhibits that from happening. So we've done that. And then finally, in order for a DNA damage response inhibitor to work, you have to have something that damages the DNA that allows for the inhibitor to have an activity. And oftentimes, you'll be combining radiation with a PARP inhibitor to create that damage. And we want to get away from radiation. So being able to have a single molecule that can not only create the damage of the DNA in the tumor, but also inhibit its repair, leading to that apoptic cell death. We're focusing on some fairly important markets: ovarian cancer, triple negative breast cancer, prostate cancer. These are tumors that have a couple of features. Number one, we know that they have a high burden of DNA damage response mutations, so they're ideal targets for drugs like this, number one. And number two, these have been indications where companies have been very successful in moving toward what's called accelerated approval with the FDA. This means that you get your drug approved based on a phase 2 study that shows you shrink the tumor, not a large phase 3 study. And that lets us move very quickly toward approvals. And this is what we're after, and one of the reasons we want to be in the public markets with access to that capital. So 3 classes of compounds. We are in lead optimization with these, with the 2000, 3000 to 4000. I'll go through these in individually toward the end of the presentation, and we're looking to move into the clinic here within about 18 months from funding. We're very excited to be in this position. How we're going to do this and how we can do it so rapidly is because we've got a great collaboration with the University of British Columbia, with a professor called Maj Dugard, who's an expert in DNA damage repair. He's got the systems in his laboratory, including the animal models, to go through the entire lead optimization process. Dennis Brown and I have worked extensively with Professor Dugard before. At a previous company, we actually asked him to do diligence on the KT 3000 and 4000 series of compounds, and his response was, wow, this is so interesting and different. Let's do this in my net lab. How can I become involved? So we've established a collaboration at the University of British Columbia to be able to do this work in his lab with that infrastructure, which saves us a ton of time and a ton of money, and it allows us to move very quickly. We also will continue to enjoy the services of Wang Shen, the lead chemist here, who's designed these compounds, so that we can have an iterative process if necessary to make incremental improvements, sort of the engineering, if you will, as we learn more about how these behave biologically. But I'll show you some data at the end here that says that, you know, we believe we have potentially lead compounds in hand now that we can just move rapidly through. The team, in addition to Dr. Dugard and Wang Shen and myself, my background is biophysics. I've been doing this for about 25 years, building companies in the biotech space, a number of companies that are now public in the US, for example. Maj Dugard has a significant history of spinning successful companies out of his lab. Dr. John Langland is someone that I've worked with for over 20 years on the operating side, managing the science, is a wonderful person there. David, as I mentioned, has joined us as Chief Financial Officer. Mike Liggett and Alfredo de la Crizia will be on the board. Mike I've known for over 20 years, and he'll be chairing the audit committee of the board. Dennis, I've worked with in a number of settings, and Len Post will be on the board and the scientific advisory board. Dr. Post was the individual who was the champion of the drug that Pfizer acquired for $14 billion at his prior company. So, wonderful person alongside of us, history of great company building, as does Dennis. Dr. Sankar, big pharma and biotech experience, a number of FDA approved cancer therapies that he's been involved developing. He's going to be our chief medical advisor. And of course, Dr. Wang Shen, the chemistry wizard behind all of this, will be intimately involved as well. So great starting team. We've already got resumes and recruiting for the hands in the lab at UBC. So this can move very, very quickly upon funding. Intellectual property, these are all molecules that are new. We own the composition of matter. So if you think of it from sort of a mining analogy, we own the real estate. And we have great engineers and geologists to understand where to drill down in that biology to move the right compound toward the right tumor in clinical trials. The issued and pending patents around the KT-2000 PARP inhibitors extend out to at least 2032, and the 3000, 4000 series extend to at least 2041. Our team has a great history of building on older molecules and extending patent life, so you can rest assured that we will be able to continue to build IP around these even beyond these long-term dates that we've got in front of us. But it's a great position to be in. Owning the molecules, owning the composition of matter is what drives partnerships with big pharma. Our financing strategy, as I said, this is a subscription receipts offering. We'll have initial cash resources with this financing, plus the existing cash in the CPC, of about $3.1 million. This will fund us well beyond 12 months. As you can see from the use of proceeds here, there's a fairly bit of unallocated capital that'll take us beyond 12 months. And the idea is that we will use the public markets opportunity to raise additional capital to fund clinical trials at about 18 to 24 months after this closing, as we anticipate moving toward clinical trials at that time. Use of proceeds, this is not going to be a quiet story as we go through the lead optimization. We'll have a number of things that we're looking to validate, continued validation in PARP-resistant tumors, which I'll show you some data here in a minute, showing the brain penetration, combination studies with immunotherapy. All of these will lead to data that can be presented at peer-reviewed scientific meetings, and we believe that's the right way to communicate success and progress as we go forward. As we move into IND enabling studies and eventually into clinical trials with additional capital raise, there'll be a constant flow of news showing progress in the science, which we believe will drive significant shareholder value. This isn't our first rodeo. We know what's important to communicate, and we know how to get the data to make that happen. So in a nutshell, what we've done is we've put together multiple programs that we believe establishes a leading presence in the field of DNA damage response inhibitors. We are targeting blockbuster cancer markets where there are unmet needs, where targeting with drugs like this makes sense, and where we have a good history of accelerated approvals at the FDA. We've got excellent IP in terms of the patents and owning the compositions of matter and uses of these molecules. We've got a great team that's done this before and can move very quickly. And we believe that this pre-money valuation here, it creates a very attractive point of entry for new investors with very significant upside and obviously near-term liquidity as things will be free trading. I'll make myself available, as will Dennis, to talk about any questions on the science or anything at all. On the financing contacts, David and the legal folks at Blake's are taking the lead there. Subscription agreements should be submitted to Blake's, and the funds will come to the company and then be immediately moved into an escrow. So I would like to walk through some science really quickly, but I'll stop right there to see if there are any questions on the business side of the company first.
So, in regards to the aggressive tumor regrowth and the increased mortality rate after, I guess, a cell is maybe able to get around the PARP inhibitor, is that a side effect, or is that something that's common?
That's pretty common. So it's not just you don't see that just with PARP inhibitors, but tyrosine kinase inhibitors, EGFR inhibitors in lung cancer, HER2 inhibitors in breast cancer, even with drugs that have become very important like Avastin. When you hold a tumor at bay, that tumor is trying to survive, and it tends to come back more aggressively than when you started once it breaks through that therapy. And so what we've done here, and you'll see this in the next few slides, is to sort of anticipate what's going to happen when that resistance begins to develop. And this is very important, and it's very new science. That's why this is so cool, is that the understandings of the resistance are evolving. And so we're taking that cutting edge knowledge and then designing molecules to take advantage of what we're knowing. So sort of kind of skating to where the puck is going to be and getting there before the tumor does and cutting it off. And so that's the next step here. And typically this is very typical iterative science. You'll have the first generation of drugs which have a great impact, and then you learn about resistance and you design the next generation of drugs and have an even bigger impact. This is just how drug development does. So it's not really a side effect. It's just the natural, the natural progression of a cancer cell that's trying to survive.
Okay. Thank you.
Okay. So let me walk through some of those real quick, and then we'll come back and answer additional questions. So as I said, we've got the three classes of compounds here. The most advanced being the 2000 series. These are novel, patented orally. So these are pills, not injections, PARP inhibitors. We're showing a structure here which has a lot of R's and Z's. That means that's just a nice way to say we're not telling you the whole structure of the molecule. And what we've done is we are targeting resistance in FDA-approved agents, so tumors that have become resistant, and particularly being able to chase the tumor into the brain. And Wang Shen is an expert at developing molecules that can be moved into the brain. So we've got the patents on these. And what you can see here is that if you look at the FDA-approved PARP inhibitors, they will have a sort of a commonality in some of the structural features that we maintain. And then we've taken all of the knowledge about their drugs and designed what we believe are better ones. You can see in terms of the potency, these numbers, the IC50s, the examples, all of our compounds pretty much hit that same range of what's called low nanomolar potency. So we're just as good at hitting the target as the FDA-approved drugs, but we've also designed features that we believe will allow the brain penetration. There's some work to do to validate this, but this is a very simple experiment here showing some chemical features where you want to have a high log P and a molecular weight below 500. And the 2000, 2207 and 2153 molecules look to have a slightly better profile than niraparib, which is the only FDA-approved drug to show good preclinical brain penetration. It hasn't really been validated clinically, but we think we're better. So we know we've got a great starting point. And we also have great data showing that these drugs work very well when they get to their target. So this is a very simple model of triple negative breast cancer, which tends to be resistant to PARP inhibitor treatment. You see here that in the vehicle, which is just basically saline, you're allowing the tumor to grow in the animal. You're treating with either your drug or nothing in the vehicle, just salt water. And you'll see that the tumor in the vehicle grows quite aggressively, and the 2125 treated with our PARP inhibitor doesn't. And if you look at that graphically on the left here, you see just over a month, that tumor with the vehicle is growing gangbusters and will kill the subject animal. But the PARP inhibitor with our treatment with ours does not show any growth at all. At the same time, we're not seeing side effects. So you would normally see if your drug is causing all sorts of side effects in the treatment animal, you'd see its weight going down. And you see there's no change in the weight of the animal versus that's being treated, whose tumor is being kept at bay and not growing and shrinking, versus an untreated animal whose tumor is growing gangbusters. So these are the most advanced compounds we have, and the brain penetration we believe is there. We'll validate that. If we need to tweak the chemistry, we've got the capability to do that, and we can move very quickly in that collaboration with UBC. The second class of compounds here, as I mentioned, we now know that one of the things that happens in a tumor cell is upregulation of secondary mechanisms such as HDAC. And so the 3000 are dual action DNA damage response inhibitors, where we're not only inhibiting the PARP functionality, but also anticipating, sort of, as I said, skating where the puck is going to be, because we know that the tumor is going to try to upregulate the HDAC mechanism, and that we want to inhibit that as well, which then improves the therapy. And these data here just are showing, if you look at the FDA-approved HDAC inhibitor or FDA-approved PARP inhibitors, our potency and anti-tumor activity is bang on in terms of molecules that are FDA approved that only have one of these functions, either PARP or HDAC inhibition. And what's the advantage here? Well, if you look at a tumor that's become resistant and you're treating it with the AstraZeneca drug olaparib, and you look at our combination drug, you're seeing a over 5 times greater potency in that resistant tumor compared to the FDA approved drug, because we have that dual functionality. So very important opportunity to move these forward rapidly. And then the final series, as I said, you need to have some sort of DNA damage to have a DNA damage response inhibitor matter. And so if you just have a cell that's doesn't have DNA damage or doesn't have a defect in a secondary repair pathway, the PARP inhibitors don't do much. And so by bringing a functionality, adding something to the molecule, which in this case is a potent methylation agent, which is part of the molecule, that actually will cause the DNA damage, which then calls the PARP inhibitor in to try to repair it, and we inhibit that, which leads to further DNA damage and cell death. So here we're looking at an opportunity to open up the field into much broader types of tumors beyond the ones that are typically targeted by PARP inhibitors. So that just gives you a little flavor. And here you were showing that the inhibition at PARP is on par with FDA approved inhibitors, but we're able to cause that DNA damage as well. So that's really just a quick overview of our science. In the full slide deck, you have the full team bios, risk factors, and obviously the other things that go along with a public listing. So with that, I will turn it over to y'all for any further questions.
Thanks so much for the presentation, Jeff. I just had a question on commercialization timeline for something like this. Just to clarify, how long do you think it realistically takes to have a drug actually being sold and making revenue?
It will take 3 to 5 years to get there, which is short in drug development land. The reason we can get there relatively quickly is because of that opportunity for accelerated approval, where you're not having to do a large, hundreds and hundreds of patient phase 3 study. If you look at the most recent PARP inhibitor approvals, those were studies of under 100 patients, in some cases under 60. And there are other examples where approvals have been granted by the FDA in tumors with an unmet need, with studies of under 30 patients. So we can move very quickly. One of the things that we'll have to do is establish enough experience in human clinical trials that we have a good understanding of the safety of the drug. So we're not going to go out and just treat 20 patients and say, hey, FDA, we want approval. We'll build a safety database of a couple of hundred patients over time, starting with the initial clinical trials, and then as you're brought into early phase 2 studies. You do that as quickly as you can, and at the same time, you identify that one tumor that you want to go after for that accelerated approval, and you design a small study to do that. So that entire process is a 3 to 5 year endeavor. We'd like to say that we're going to be on the short end of that timeline, but obviously time will tell. This is science, and you have to be flexible and build on what you learn. If we have showing great brain penetration with the drugs that we have in hand now, that 2000 series will be moving into pivotal preclinical studies and clinical development very quickly. But if it takes more work, then it might take a little longer. But to answer your question, 3 to 5 years. Now, having said that, the opportunity for liquidity for shareholders, because we will be public, people can take an upside profit at any time, number one. And number two, when I pointed out those multi-billion-dollar takeouts of companies with PARP inhibitors, those were all prior to approval. So, the likelihood is if we have, something as cool as we know we do, there will be opportunities for potentially licensing or even having the company acquired for, very, very substantial return from where we're starting prior to commercialization.
Great. Thanks so much for sharing that.
Jeff, maybe you can give us a lowdown on where the going public process stands and how that will play itself out.
Yes. So let me go back to the offering summary here. So as I said, we've got over $1 million committed so far, including insiders. So we are accepting subscription receipts and our subscriptions in the offering as we stand. We're hoping to complete this non-brokered private placement before Christmas. The filing statement has gone into the exchange, so they are in the process of reviewing that right now. The audit of the financial statements is going on in parallel with that. That should be done in the next week or so. So, assuming we can close this offering on time, ideally before Christmas, I'd expect the exchange to be through their process in January, and or hopefully early January, or even if we're lucky, maybe by the end of the year. But let's say January. At that point, the transaction, the merger with Vincero Capital closes. The company is listed on the Toronto Venture TSX Venture Exchange, and the investors in this round will have freely trading shares and the warrant as well, and the share underlying the warrant is free trading as well. So we've been working on this for a few months, and you can see that the deal that we signed with Vincero was back in August. Since August, we have set up the company, put together all the exchange filings, and also acquired the rights to the 3000 and 4000 series. So we've been actively operating the business as if we're moving this all forward already, but obviously we need the capital to really go.
That's great.
Yeah, I have one more question. Is this effective on genetic forms of cancer as well too, or just I guess cancers where your DNA has kind of been damaged? What was it called? Sorry, DDR mutations.
Right. So the answer to that is generally yes. So one of the, when you talk about a genetic form of cancer, for example, a ovarian or breast cancer patient that has a genetic BRCA mutation, okay, that genetic underlying feature of the cancer is a DNA damage repair mutation. So it's the genetics that are underlying it that leads to that mistake, that leads to the cancer, that leads to the opportunity that we're targeting. So the answer is generally yes.
Actually, I got one other question for you. So the manufacture of these molecules and the modifications as you're refining it, do you have a CMC partner in place, and what's the cost of that process? Is it
right? So the way that we manufacture the molecules for the experiments that will be happening at UBC, we have a small chemistry laboratory capability with Wang Shen that can make all these things. When we get to the point of scaling up, to move something into human clinical trials, that's going to require a different, sort of different manufacturing capability that we have in-house. But there are contract manufacturers that are experts in the cancer field. You'll be looking at an investment of in the hundreds of thousands of dollars up to maybe about $1 million, depending on how complicated the manufacturing is. But we've done this before. We have relationships with those well-known manufacturers. So this is, once you get to the point of needing them, they're available. We always look at the cost of manufacturing from the earliest stages. So we wouldn't bring a molecule forward that's going to be incredibly complicated to make or scale up. That just wouldn't be possible to have as a product. So we keep that in mind. We'll be able to do it in-house for the next steps of lead optimization and development, but eventually we'll contract that out.
That's great. Excellent.
Another question, what jurisdictions are your patents granted in? Are they just North America?
Global. We own the worldwide rights to everything with the exception of the 2000 PARP inhibitors, where the Chinese rights have been retained by the company that funded the original development, called Kenyan. So if you go back to the history slide here, back in 2011, Kenyan funded Wang Shen's work, and they retained the Chinese rights to the 2000 series. Everything else, the 3000, 4000 series, were invented independently by Dr. Shen. So we have the global rights. We'll have global rights to those.
Just one more quick question, Jeff. Do you want to just touch on the accelerated approval process again, and how you're a candidate for that?
Right.
Just touch on it quickly.
Yeah. So a few years ago, the FDA adopted some regulations which are known as accelerated approval. It's 21 CFR Part 3, 14 subpart H is the law itself. And what this does is it says that a drug developer focusing on a serious and life-threatening disease where there are no available therapies can be granted approval on what's called a surrogate endpoint. In cancer, the gold standard of what you're trying to do is make the patient live longer than the existing available therapies. But the FDA has accepted, and everybody agrees, that a good surrogate for an extension of life is showing that you shrink the tumor. So if you're making the tumor smaller or making it go away, the patient's not going to die from cancer. End of story. We all accept that. So you can design a study that says, I shrink the tumor. And you don't have to compare your drug to anything else in that case. You just have to show you shrink the tumor. And that's where, as long as you have a big enough, big enough number of patients already in the sort of safety database, you can show in a relatively small population for a specific tumor, often an orphan drug indication, if you will, that you can go forward, shrink the tumor, and get that accelerated approval. So if you look at the PARP inhibitors, generally we're all approved on an accelerated basis, which is, now these are all becoming blockbuster products, and the studies were generally under 100 patients. And even if you look in other indications like lung cancer, a drug called osimertinib was approved in a relatively small study in 2015, and is now doing about $2 billion a year in sales, because they've grown that indication from the initial approval through other studies. So what you're doing here is identifying the right tumor, showing you can shrink the tumor, and seeking your accelerated approval, and then building the market from there. And this is a very effective way to move rapidly through the FDA process. It's something that's been put in place because it's been recognized that we need more drugs to treat cancer. And we are building our portfolio toward tumors that we can take advantage of the regulations that are in place to move as quickly as possible.
Awesome. Thanks for sharing that. And just one more quick question. So if a patient has is diagnosed with cancer, can they request this kind of drug, or do they have to first exhaust all other options?
So that's interesting. When the drug's approved, doctors can use it for whatever they believe it's going to be effective for. So in that case, the patient could kind of say, hey, I read something here and I'd like to try this, and the doctor can agree with them and prescribe it, even if it's not approved there. In the development of a drug, generally you're going to focus on patients where you believe you have the strongest chance of success. Having said that, there are certainly instances where patients or doctors have said, there's a drug in clinical trials. It's not being studied for the cancer my patient has, but I think it's going to be valuable there, and I would like to try it. And then at that point, it becomes up to the FDA and the company, the sponsor of the drug, to determine whether or not they agree that the science is supportive, and then that patient can be granted opportunity to get the drug on a compassionate use basis. Sometimes this leads to new understandings and a new opportunity, but we believe and have always in our prior companies supported clinicians who have been interested in testing the drug or using the drug in a patient where it's outside of our field study, as long as the science makes sense. Our job is, number one, to build shareholder value by getting something across the goal line as quickly as possible. So we're going to be laser focused on that. But to the extent that there are things around the edges that are scientifically interesting, that a doctor wants to move forward because they believe it can help their patient, we'd certainly support that by making the drug available. We wouldn't put any money into it, but we'd make the drug available to that doctor.
Great, thank you.
Good. Well, I think that sounds like we've come to the end of questions. So at this stage, if we can, thanks a lot, Jeff, for your presentation.
Very good. Thanks, guys. And really appreciate everybody's interest and attention. And, Sean, when you send around the recording, obviously you'll include mine and Dennis's contact information there as well. So if and David, so if there's any questions at all, we're easy to find. Very good. Thanks, everybody.
Thank you very much.